It can invade both reticulocytes and matured erythrocytes. So all cells, all red blood cells rather, at different stages of maturation can be invaded by the P. falciparum parasite. As a result, the resulting anemia is devastating. Whereas with other strains such as Vivax and malaria, they have particular subsets of red cells that are invaded. As a result, the degree of anemia is not as bad as what you have with P. falciparum. Now, because these organisms are microaerophils, you know what that means? Microaerophilic organisms, what does that mean? Do you know the exact condition? Do you know the exact condition that we describe as microaerophilic? No? Okay, so don't worry. Because they are microaerophils, schizogony tends to occur at the deeper level. So in deep tissue, you have schizogony taking place. Now, like I told you, once the red cells are invaded by the parasite, they become relatively more sticky. And when you have them in deep vessels that are very tiny and they get stuck together, they are going to occlude the vessels. So you can also have evidence of ischemic injury to organs such as the kidney, such as the brain, depending on where you have these parasite cells lodging in. Now, apart from that, P. vivax and P. ovale are known to produce dormant liver stages. These are referred to as hypnozoites. Now, the significance of these stages is that once you treat a patient who has obvious infection and you don't take care of the liver stages, the patient may have a recurrence because these hypnozoites may revert to forming schizons and merozoites. Okay? Now, in terms of epidemiology, P. falciparum used to be worldwide in occurrence, but in some temperate areas of the world, they have effectively controlled malaria parasite infection. In Africa, it's the predominant strain. P. vivax is mostly found in Asia, Latin America, and in some parts of Africa. In West Africa, P. vivax is not common because the receptor required for parasite entry into the red cell is lacking in the cells of people of West African descent. P. ovale is found mostly in Africa, especially West Africa. Some people believe because you don't have vivax infection, P. ovale takes over. It's biologically and morphologically similar to P. vivax. Now, P. malaria is also found worldwide. What I want you to note about this particular species is that it's associated with nephrotic syndrome in children. And like I said, P. nolensi is the new addition to the list, and it's common in Southeast Asian countries. So now we've gone through the life cycle. What, I shouldn't have put the slide up first. I should have asked you what sample you're going to send to the lab. So what sample are you going to send to the lab? Blood. Why? Because it's a blood parasite, yes. Okay. So what are the approaches? The gold standard is to perform what we call a blood smear. Now, what does a blood smear mean? It means taking the patient sample, which could be peripheral, venous blood, and making preparations that we call thin or thick film. You'll learn more about this in the lab, but let me just quickly put you through. Now you see here, this is the blood film preparation, and this is another blood film preparation. What are the differences that you can observe? Is that all?
No, so it's really on the mic.
Ah, okay. So just anybody, what can you appreciate the difference between these two? Just a guess. You can't be wrong. Don't worry.
Cells are more clear on the mic.
Which cell?
The erythrocytes.
Very good. On this preparation, you can appreciate the erythrocyte, and you can appreciate that there is anemia. You can see that the central pallor is quite pronounced, right? And here, you cannot appreciate any red cell, right? Very good. So this is a thick film preparation. This is a thin film preparation. And what we do essentially is this. While preparing a thick film, when we take the patient sample, we mechanically break down the red cells. So we actually mix it on the slides to break down the red blood cells and release the parasites if they are present. Whereas in a thin smear, what we do is gently spread out the blood sample on the glass slide and we fix it so that the red cells are not destroyed. Why do we do this? We want to be able to see the parasite within the red cell. Because when we do that, we can, one, confirm the diagnosis. Two, we can also give an idea of the species that is causing infection. Remember we said P. falciparum is the most dangerous out of all these strains. So you want to be sure that the patient doesn't have P. falciparum or has P. falciparum so that you can be very aggressive in therapy. So what are the things that we can see? If we focus here, you can appreciate that you have some bluish structures within the red cells, right? Now these are the parasites, the trophozoite stage. If you look at this red blood cell, you can appreciate two parasites, right? Here you can appreciate about three. When you see features like that, it's highly suggestive of P. falciparum infection. You tend to have multiple parasites invading one red blood cell. Is that clear? Now, apart from that, yes. Okay. Like I said, I prefer pointing out. I didn't come with my pointer, I'm sorry. Okay, so what I'm saying is this. These smears that are on the right, the upper and the lower right micrographs, they are showing thin film preparation because we can see the red blood cells in the film. Whereas the micrograph in the middle, the lower micrograph in the middle shows a thick preparation because the red cells have been mechanically destroyed. Okay? Now, the reason why we prepare thin films is because one, we want to make a diagnosis, right? We can see the parasite in the red cells. And two, we want to know the species of parasites we are dealing with because we know P. falciparum is a dangerous species and we must treat aggressively. Now, what I was trying to explain is that when you look at the number of parasites that you can find within a red blood cell, and you observe multiple parasites in a red cell, as you can see here, you have 1, 2 parasites. Here you have like 3 parasites. That is highly suggestive of P. falciparum, okay? Another feature that may suggest that you're dealing with P. falciparum is that you have parasites that appear to lie on the margin of the red blood cell. So it's like you have this parasite closely associated with the margin of the red blood cell. So this again is an indication that this is likely P. falciparum, okay? Apart from that, if you look at the parasite morphology, let me see, I'm not sure. Okay, yeah. If you look at this red blood cell, you can see it's like you have 2 chromatin dots, looking like the head fold. So that's another feature suggestive of P. falciparum. So when we look out for the headphone morphology, multiple parasites, parasites that are lying on the red cell membrane. All these are indicative of P. falciparum infection. Is that clear?
They want us to use the phone because we can't see.
Yeah, I was trying to move the mouse. Okay, thank you.
And look to the microphone. So if you move away, they can see. Thank you.
Sorry. I'm sorry. Okay, so for the benefit of those who are online, I'll go over the explanation again. Like I said earlier, these two micrographs are showing thin film preparations because the red cells are preserved. This particular film is showing a thick film preparation because the red cells have been mechanically destroyed in the preparation of the film. Now, the features we see on thin blood smear that can help us to make a diagnosis of P. falciparum includes the following: having multiple parasites in a red cell, having parasites that appear to lie on the red cell membrane. You have one here, okay? And yeah, this is another one. And having parasites that have double chromatin pattern, what we refer to as the headphone appearance. All these are highly suggestive of P. falciparum infection. Another feature that you can look out for are the gametocytes, if you get to see them. Here, you have the gametocytes of P. falciparum because it's the only species that gives the crescent-shaped gametocyte. Other species gives roundish gametocytes. So when we see this banana-shaped or crescent moon-shaped gametocyte, it is highly diagnostic of P. falciparum infection. So that's as far as blood smears go. We also have what we call rapid tests, in which you have antigens that are associated with the parasite, immobilized on a nitrocellulose strip. So what you have here in the rapid test, you have what we call the parasite lactate dehydrogenase. It's an enzyme that is common to all Plasmodium species. With P. falciparum, we have what we call the histidine-rich protein 2. It is highly associated with P. falciparum. So what you have here is a strip that contains these antigens, and you have a control antibody. So if a patient is infected, antibodies are produced against these antigens. So when you introduce the patient sample, the likely results that you can have are the following. The black line suggests a positive reaction, okay? So here, you have only the control line showing a reaction. So this means your patient sample is free of any antibodies against these antigens. So this is a negative result. Are you following? Here, you have only the parasite lactose dehydrogenase line positive. Remember, I said this is common to all of them. So this has not helped you to differentiate whether you have P. falciparum or P. ovale or whatever. What it's just telling you is that this is positive. So it could be ovale, it could be malarial, it could be any other one. Then the third strip shows the line positive for histidine-rich protein 2, which is highly associated with P. falciparum, and we have the lactate dehydrogenase positive as well, and then we have the control line positive. So this is telling us it could be P. falciparum or it could be a mixed infection. It could be P. falciparum because P. falciparum will produce the lactate dehydrogenase as well. So having the two lines positive is telling you you have P. falciparum possibly as the only infective species, or you could have a mixed infection. Now, the last strip is regarded as invalid because the control line is not positive. So something must be wrong with the assay. Remember that your control line is to help you to guide you in your interpretation. So here I've explained the basis of the rapid test, and here I've included the micrograph showing you what a positive strip looks like. So here you can see the thick film. You can see a lot of the headphone trophozoites. You can see one is here, okay? So this is likely to be an infection due to P. falciparum, but you can't speciate on thick film. I'm not sure. I can only report this as malaria parasite seen. I cannot say if it's truly P. falciparum or not. But if I have the thin film preparation, I can categorically report that P. falciparum parasite seen. So the next parasite we're going to discuss, so yeah, Leishmania. Just like with P. falciparum, you have different species that could cause infection in humans. But unlike P. falciparum and the others, Leishmania species tend to cause a spectrum of infection. Some conditions are limited to just the skin, so we talk of cutaneous infection, while some can involve the skin and the mucous membrane, so we say mucocutaneous, and you can have those that actually go down for organs like the liver, the bone marrow, so we talk of visceral leishmaniasis as well. But irrespective of the presentation, the transmission is the same. You need an insect vector. In this case, it's the sandfly. As a result, the organism has an indirect life cycle, but luckily it's not a complex life cycle like that of the Plasmodium, okay? So the sandflies that are involved in transmission, two species are commonly associated with transmission, the phlebotomos and the Lutzomyia sandfly. Now, they are referred to as Old World, New World. Now, the reason is because these vectors appear to be well adapted to certain regions of the world. So phlebotomous sandflies are associated with the Middle East and part of North Africa. So they are regarded as Old World. Whereas the Lutzomyia sandfly is associated with Central America, South America, so they are referred to as the New World. But irrespective of whether it's phlebotomous or Lutzomyia, they can transmit the infection. What is the life cycle like? Again, it's the female species that comes for the blood meal. They need the heme for their eggs to mature. In this case, the form of the parasite that is infective to humans is the promastigotes, not sporozoites. So the names are becoming more confusing, right? Okay. So the promastigotes are motile, they have flagellum, and once they are inoculated into the skin, they are mopped up by the macrophages. Unfortunately, the macrophages cannot destroy the parasites because the parasites prevent the fusion of the lysosomes with the phagosomes. So instead of the macrophages destroying these parasites, they actually serve as the source of nutrients. So the parasite changes form and becomes non-motile. So we refer to them as amastigotes because they don't have flagella again. They are no longer motile. And within the macrophages, they start to multiply. Eventually, they destroy the macrophages and then they invade surrounding macrophages. And that's how the infection progresses. When an uninfected sandfly comes for a blood meal, it's going to pick up the macrophages, and within the sandfly, the macrophages becomes the procyclic promastigote. It undergoes different stages, which I'm not going to bother you with. Eventually, it becomes the promastigote, stays in the salivary gland, When that infected sandfly goes for a blood meal, it inoculates the organism into the bloodstream. Okay, so in terms of the pathogenesis, what the organism does is to invade macrophages, like we said. Eventually, particularly with visceral leishmaniasis, there is reduced activity of the bone marrow. And you know what the bone marrow does. So eventually you have the patient presenting with anemia, there is decreased white cell count, and there is decreased platelet count as well. So patient may present with bleeding, recurrent infection, anemia, and things like that. Okay, so I've mentioned this, that you have a spectrum of infection. You can have infection localized to the skin. You can have involvement of the mucous membrane or the deep organs in visceral leishmaniasis. So how do you make a diagnosis? Should have asked this question before putting the slide up. What samples can you send to the lab? Remember, you can have cutaneous lesions. So you can send in samples from the skin biopsy, fluid that you extracted from the margins of the lesions on the skin. You can send all these to the lab. In patients presenting with visceral leishmaniasis, it's better to send in a bone marrow aspirate or a biopsy, depending on how good you are at retrieving these samples. Excuse me. You can also send in blood for culture, okay? And when we culture, the form that we see are the promastigotes, okay? So if we see the promastigotes growing from the sample you send to the lab, it's diagnostic. We can also look out for specific genetic materials using molecular methods. And since there are parasite-specific antigens to which the body produces antibody, serology may also help us in diagnosis. Now, when we prepare a smear of the bone marrow or the skin biopsy or aspirate, what are we looking out for? We are looking out for macrophages that contain the actively dividing parasite, the A mastigote. And this is what you can see here. The arrow points to one macrophage that contains a lot of parasites. Okay, so this is diagnostic of Leishmania infection. Another parasite that we are discussing is Trypanosoma cruzi. This is the cause of American trypanosomiasis. Now, unlike the two parasites we've just discussed, that is Plasmodium and Leishmania, which are transmitted by inoculation when the vector comes from a blood meal, the Rudivig bug is a different vector in terms of how it behaves. It's also referred to as the kissing bug because it tends to feed at night and it tends to come for the face because of CO2 production. So the part of the body that has the highest CO2 concentration, that's where the bug will move towards. And what this bug does, because it's an avid blood sucker, when it starts feeding on blood, it defecates so that it can widen his abdominal cavity to take in more blood, okay? The infective form of the parasite is found in the feces of the Rudivig bug. So it's not the bite that transmits the infective form. So when the bug bites, the bite is painful. So as a reflex, the person being bitten is going to rub in the feces into the sore. So it's not really true inoculation, okay? But you're going to rub it in. So that's how it's transmitted. Other routes through which transmission could occur include blood transfusion, organ transplantation from a pregnant mother to the unborn child. And when you work in the laboratory with this organism and you're not careful, you can actually inoculate it and cause infection. So what is the life cycle like? Now, like I said, the triatomy bug feeds at night. It comes, it bites, and in the process, it's going to defecate. Now, in the feces, you have the infective form, the trichomastigotes. These trichomastigotes are going to be rubbed in, okay? And once they have access to the blood circulation, they invade different cells, okay? They can invade the macrophages. And once they do that, they change form to the amastigotes, just like with Leishmania species. Now, what they do, which is quite different from what happens with Leishmania, is that the amastigotes, when they are released, they can revert to the trypanosoma stage, that is the flagellate stage, and this can invade new cells, and the infection can continue. An uninfected reduvic that comes for a blood meal can pick up these trichomastigotes, and these are going to undergo further division and maturation to become what we call the metacyclic trichomastigotes, which is the infective form in this newly infected triatomy bug. And when this newly infected triatomy bug goes for the blood meal, the life cycle continues. So how can you make a diagnosis? In the acute phase, getting the blood sample can help. We can see the trichomastigote in blood before it invades cells and become the amastigotes. If you get the patient in the chronic phase, serology may help, or you collect a biopsy and we look out for the amastigotes in tissue cells, okay? So I have to rush now so that I can finish. Another strain of trypanosoma species that can infect humans are Trypanosoma brucei gambiense and rhodiense. Now, the brucei in the name is to honor the guy who did extensive work with this parasite and was the first person to report the infective form in cattle. So that's a bruise. So Trypanosoma brucei gambiense and rhodiense are responsible for sleeping sickness. They are not responsible for American trypanosomiasis. That is due to Trypanosoma brucei, okay? The morphology and life cycle is quite different from that of Trypanosoma cruzi, because the insect vector in this stage, in this parasite, is the tsetse fly, not the reduvid bug, okay? When the tsetse fly comes in for a blood meal, it's going to inoculate the metacyclic trichomastigote. Remember that with T. cruzi, the organism, the infective form is not inoculated. It is passed out in the feces, and then the host is going to rub it in. But with Trypanosoma rhodiense and gambiense, the infected tsetse fly actually inoculates the trichomastigote. Now, unlike Trypanosoma cruzi, it does not invade the macrophages or tissues, so there is no amastigote stage. It maintains a purely extracellular life cycle, dividing by binary fission, as you can see here. When an uninfected tsetse fly comes for a blood meal, it's going to pick up the trichomastigote, and once inside the tsetse fly, it's going to undergo further development and eventually becomes the metacyclic trichomastigote, which is the infective form. Now, with this parasite, what sample are you going to send to the lab for diagnosis? The skin won't help. Blood, yes. So we can send in pathogenesis. This is self-explanatory. I'm going to quickly round up. So blood sample, and you can actually demonstrate the trichomastigote in blood smears, okay? And this will be a thin smear because you can see the red blood cell, okay? Now, how can we prevent and control? All the parasites we have discussed are vector-borne, right? If you control the vector, you prevent infection. So how can you control infection? vector, sorry. With malaria, there was recently this suggestion that they are going to release genetically modified male mosquitoes in one state in the US. And there was a lot of people complaining it's unethical and things like that. Now, the reason is this. The female Anopheles mosquito mates only once in her entire lifetime. So if you are able to genetically make male mosquitoes that are not sterile, she doesn't know that the male is not sterile. She mates only once. So her eggs will never mature if you release genetically modified sterile males into the environment. But people are worried that you just don't know. These genetically modified males may start transmitting something else because they're not normal. So anyway, but that's the idea. The female mosquito mates only once. If the male is sterile, that's it. Our eggs will never mature. Another thing that you can do is to destroy the breeding sites of the vectors. So there are some species of fish that can feed on the larval form of malaria of the mosquito vector. What you can also do is that you clear vegetations that surround the house. If you have open cans that collect water, make sure that you empty the cans and destroy anything that can hold on to water that can encourage breeding. Then you can prevent vector bites, particularly if you're going from a non-endemic area to an endemic area. You're traveling to Southeast Asia, you're traveling to Africa for whatever reason. Pack long sleeve tops and long pants. This is not the time to wear short pants and crop pants, okay? You can also use insect repellents that won't allow insects to bite you, okay? And with particular reference to Trypanosoma cruzi, it's been known that houses that are built of mud with thatched roofs can encourage these organisms to breed. They hide in there and they breed, and then they come out at night to feed. So if you improve the housing, you have concrete housing with corrugated iron roofs and things like that, that does not encourage the breeding of the vector. So thereby you can prevent infection. Another thing that you can do, which is not on the list, is that when you get an index patient, you must treat aggressively. Because remember, humans are the reservoir for malaria parasites. Without humans, the life cycle will, if you don't have gametocytes there, the life cycle ends once that person is defectively treated. So you treat in areas where you have P. vivax and P. ovale endemicity, you must treat for hypnozoites. Because if you don't, these are the forms that can revert and cause the recurrence. So these are the things that you can, put into your control plan, sleeping under mosquito nets and things like that. So any questions? Let me check the chat box. Any questions? Okay. Where's the chat box?